Hello everyone. Today we will be talking about plant tissue culture. So, as you can see here, basically, if you see the pictures over there, it's just like we are taking care of a baby inside a test tube. So, similarly, in case of plant tissue culture, instead of baby, the plants are being cultured. So, let's see what is plant tissue culture. Now, basically, if you see, it's a process of growing cells artificially in the laboratory. Now, one thing is very important that when you grow something in a laboratory, especially in plant tissue culture, you need to take care of aseptic conditions. So, what does we mean by aseptic conditions? When we say that aseptic conditions, basically we are talking about a very sterile environment. And obviously, a plant has to grow in this sterile environment, so it needs food. And that food is basically the different types of plant tissue culture medium that we will be providing. So, yeah, let's see a little bit of history about plant tissue culture. So it had its origin in 20th century, and as you can see in the picture, G. Haberland, he was the first person to work on that, and he is also known as father of plant tissue culture. So coming to this part is very important. This explant collection. So explant basically means, if you see the word, it's a combination of two words, ex and plant. So ex means excised, excised. So from a mother plant, any part which is excised, that is your explant. So here I've given you a picture where you can see apical bud can form a part of explant. Leaf also can form a part of explant, axillary bud, internode, and roots. So these are the different types of explants that we can get from a mother plant. Even in the pictures on the right, seed also can be used as a source of explant. Now one thing is very important when we go for the selection of an explant. The part of the plant is very important. Which part are we taking into for our plant tissue culture works? Usually the apical buds of meristematic zones are very good and they are free of any contamination. Now physiological condition also you have to take care, then season, collection period, then position on the plant, and the size of the explant. Now in plant tissue culture, it is found that smaller the size of the explant, the better is the response. Now coming to establishment of the explant in a culture medium. So for this multiplication, different stages are there where we use different types of medium. Basically cytokinin and auxin are used. So cytokinin is basically for shooting, that is formation of shoots, whereas for auxin it is the root. It is basically for rooting. Now once you do the multiplication, callus is basically a mass of undifferentiated cells that also will show you all in the subsequent slides. Then root formation is there. Once the plant has a shoot and a well developed root system, then we can go for acclimatization. That means you are slowly bringing out the plant from the lab condition to a, basically we are talking about taking out from the sterile environment to a contaminated environment, that is controlled contamination is there. Then finally hardening before it is released to in the wild. So these are some of the pictures where you can see different types of medium or media. One of the most common and the basal media used in plant tissue culture is Murashik and Skook's media, or commonly called as MS media. Basically it's an MS medium. Now there are different plants, they require different types of medium. Here I'm not discussing. In subsequent maybe lectures we can take up those topics. Now this is washing room. So basically plant tissue culture, what are the requirements? So you can see here, this is the washing room, which is very important, as we as I've told you earlier also, we need a sterile environment. So it starts with the washing phase. So here all the glasswares that will be used for plant tissue culture will be subjected to washing. Coming to the media preparation room, here you can see the picture, two pictures are there. In one of these pictures towards your left, you can see two research scholars, rather I should say, two students. They are working, they are preparing the medium they are preparing, in this case is MS medium. On the picture on the right, you can see a girl is there. She is basically discussing or training the students about telling the importance and how to prepare MS medium. Coming to the autoclaving, so once the medium has been prepared, it has to be autoclaved. Remember, we are talking about in a sterile environment. So basically what we are doing, we are putting it in an autoclave and we subject it for 15 minutes of autoclave, 15 to 20 minutes basically at 50 psi, and the temperature is 121 degree centigrade. This is very very important to kill any germs or any microbes present in the medium. Now one thing is very very important, I keep stressing that it's basically to get rid of any contamination. In plant tissue culture, the biggest threat is your contamination, so you have to be very very careful while working in this environment. Now once the medium is prepared, we go for slant culture. So you can see here in the slide, the slant culture is there. So basically slant is prepared to increase the surface area for the explant. So remember the explant that we took from the mother plant, that will be cultured or inoculated over here. So even agar plate is there, jam bottles. So these are different ways depending on the requirement. Now coming to the cleaning and sterilization of the materials. So here I am showing you basically the seed pod of an orchid which has to be sterilized. So basically common sterilization are sodium hypochlorite is used, mercury chlorides are used. So we usually avoid even ethanol, 70% ethanol is also used for sterilization, but we usually avoid mercury chloride because that is toxic to plants. So once the sterilization process is done, then you see the inoculation is being carried out in a laminar flow. So inoculation and maintenance of culture. So sterile materials remain protected from contamination. So when you work in a laminar flow, air is there are HEPA filters are there which purifies the air, and air blows towards you. That means there is no scope for any microbes from you when you're working. It cannot enter the laminar flow. So once this is done, the plants has to be, and once the inoculation is done, then the plants has to be transferred to a growth room. So here I'll show you after 3 weeks of inoculation what is the response. So here you can see, if you see the first slide here, you can see this is the explant. The thinner and smaller, the better is the result. Please keep that in mind. Smaller and thinner, the better is the result. Now here you can see some kind of an initiation in the second one, and third one you can see I've tried to show some callus as well as multiplication of the culture. Whatever we have cultured, so from the explant there is a multiplication. Okay, so here you can see there is a proliferation, so we separated them out. If I go back here, we have separated out from the third slide, and then we started growing individual plants. So here you can see in the first one on the top, you can see one plant is growing. However, in the second one, you can see still a lot of plants are, lot of multiplication is going on. So that is the biggest advantage in plant tissue culture, multiplication of plants. Now in jam bottle, you can see they are growing well, and there we have provided auxin and cytokinin. Then you can see here once when the cytokinin was balanced, there was shoot formation, and once the shoot formation was over, we started working on with the auxin for the root formation. And if you see here in this jam bottle, you'll see that rooting also has been developed, and it is quite good rooting is there. Now this is the growth room where you have to maintain your culture, and this room is it's just like when you have seen the first picture or the first slide. You have seen that I was giving an analogy of a baby inside a test tube with a plant inside a test tube. So basically you are giving the best of the conditions. So in this room, the plants after inoculation, they are given the best condition, that is best temperature, light, as well as humidity, which is required for an optimum growth. So This growth room, basically temperature is 24 to 25 degrees centigrade and 70% humidity is maintained. And photo period, it depends on the type of plants that you are using for your culture experiments. Okay, once the plants are good enough or matured with good roots and shoots, they are ready to be acclimatized. So for this, what we do is that we take a compost, which is a mixture of charcoal, leaf litter, then moss, depending on your uh, plant material. Suppose you're working with an orchid, you don't need mud because these are epiphytes. But if you're working with a terrestrial plant, then obviously you'll include mud also in this compost mixture. Now here you can see this is the greenhouse where the plants are being transferred. So first picture jam bottle, from jam bottle it goes out to those thermocol cups, and from there you can see that picture they are growing very well. And in this net house, proper lighting, air as well as sprinkler should be there so that the water is regularly, so that the plant is regularly watered. Now one thing is important, because tissue culture plants, they are not exposed to harsh conditions, so their leaves usually lack cuticle. And if their leaves lack cuticle, that means most of the time they are in a good environment, sterile environment, so their stomatas and everything remain open for a longer duration. Now if you don't provide them with sufficient water, then in the due course of time they will die off due to dehydration. Okay, let us see some of the advantages. Creates a large number of plants from an explant. If you have seen, just from one single explant, we have cultured, or we have come up with so many number of plants. Time period is less and it can be grown any time of the year. The rapid propagation is possible in those species where seeds do not germinate. So to overcome any seed germination, you can use this particular method. Now sterilized plants are always preferred for exchange programs and all, so you always get a sterile plants. Now coming to the applications, so you can see plant tissue culture in industry, micropropagation, the secondary metabolites. So these are secondary metabolites, alkaloids, saponins, steroids. You'll see these are basically secondary metabolites are basically plant molecules, or rather I should say these are very important from medicinal point of view. If you see, these are the components which are used for different medicines and all, so you can get a lot of secondary metabolites. So a production of anther or pollen culture, haploid and homozygous line, then vegetatively propagated plants. Now you can do basically any plant that you want can be tissue cultured, but our target is any plant which is nearing or facing threat to extinction, or threatened plants or endangered, they are to be taken care of. So this technique can be very handy and is very helpful to overcome such barriers. So thank you very much for listening to this lecture.